He just walked by. He
Okay.
has collaborators from Princeton who are applying voltages across xenobots to see if they can orient to those cues. And he's currently setting up the machines with them and helping them out because they're only here for 1 week.
Okay. I'll talk about the what I did, and then people who are in at Tufts can relay this to him because this is something we had been talking about. So, um,
Thomas, I'm recording this, so I'll just circulate it to everyone, including Puck.
oh, cool. Yes, we live in the future. That's great. Um, so for those of you who were here last week, we had a really cool presentation from Pi about some kind of biological black magic that they can do to these due to the xenobots that disrupt cell-to-cell communication in some way. And the result was that the xenobots, instead of forming nice spheroid kind of shapes, they instead become strange and lumpy. And so what Pi asked me to do was to, let's see, where is it? Try to find some way to quantify this notion of relative xenobot lumpiness. Okay. And so after emailing with Pi and Mike and Josh, I sort of came to this idea that we were going to use this idea of shape entropy. All right. And so this is a wee bit technical, but I got a bunch of images of the xenobots from Pi. You all saw those last week, so I won't share them again here. But what I did was I then went through and extracted basically just a mask of the outline of the xenobot, found the centroid, and then computed the distance from every pixel in the mask to the centroid. Okay, and so that defines a distribution that I can then just normalize into a probability distribution and compute, basic entropy measure on it. And so the idea is that if the xenobot is generally pretty circular, like this is a control xenobot, the entropy should be low because the distance from the centroid to any part of the edge is about the same. And there are some corrections and whatnot that I had to build in to make sure that everything was fair. But so you can see that there's, this is a control xenobot, pretty spheroid, the distances range from, 0.7 to 1. Everything is normalized by the max distance. And then if I go to one of the mutant xenobots that Pi had done whatever he did to, you can see that like this thing is much lumpier and the range of distances from centroid to perimeter is much wider. And this is reflected in the difference in the shape entropy. I don't know if you guys can see up here, but here we're looking, the mutant bot has a shape entropy about 5 bits. The control bot has an entropy of less than 4 bits. So clearly, this measure is behaving the way I expected. And then just this morning, I ran all of the data that Pi sent me through, where is my spider? Yes, I ran all of the data that Pi sent me through a pipeline, and we have control shape entropies and mutant shape entropies. You pipe those into a statistical significance test and you get p-value of 0.00069, less than 0.05. It must be true, ordained by God. And that's sort of what I've got in terms of results. But the takeaway is that this effect is clearly real. You can look at the pictures and like, yes, the mutant bots are lumpy. You almost don't need to go through the rigmarole of computing a measure. But once you have a measure, there's all kinds of interesting correlations we could look at. So we could look at how does this change over the course of 7 days. We could look at some of the functional connectivity stuff that Caitlin and I had looked at. Do these changes in cell-cell signaling do they trigger changes that are observable in functional connectivity before the morphology begins to change? So can we start to make a timeline of morphological development, that incorporates both like the statistical and the geometric properties of this stuff? You know, that's sort of what I've got, but I'm pretty excited about it. So, if we have more data, I would be totally happy to pipe more Xenobots through this thing.
So I have old data that we never publish, which is if you make these xenobots, they're basically skin organoids, just skin. So we call the epidermis, the outer layer. Any amount of sort of internal signaling or meso tissue that's present causes a progressive amount of elongation. There's some really good published work on this, so we could probably mine that. But you can get I have a picture up in the background. Like if you take it to the extreme, you can start to get things that look like pretzels. They will actually elongate to the point that they start twisting and basically forming these super long. So if you have them restrained, they just form super long snakes and get like progressively infinitely thin. But because of the pressure, they sort of twist in on themselves like a pretzel, and you can get intermediates. So this isn't my discovery. I just replicated some work, but we might want to look at as sort of a null is the state space that's being generated by this manipulation mimicking things that are existing in the literature, or is this a new state space that we've never observed before? Basically, like, are we moving out of the realms of what's been documented to some sort of new anatomy or geometry? I think Thomas is frozen.
She is. So while we're waiting for him to return, so that's awesome, Doug. So well, I guess we can have Thomas look at this to see what is the actual data from the literature. Like, can you get sufficient data to actually compute these statistics? And then, yeah, how can you push outside those boundaries? That would be interesting.
I assume the point, I guess I shouldn't assume. It seems like a cool thing for you guys would be downstream for the MomBot or whatever, if you could create an atlas of shape space for different permutations, the AI could potentially act on that to say, if you get a little bit of A and sprinkle in a little bit of C and a little bit of F, you might get some non-intuitive thing, right? Like maybe this is a cool way to predict functional shape from sort of just base data. We just keep feeding in data into the algorithm and enriching it and let machine learning go to work on it.
Yeah, absolutely. I mean, I remember from all the early days of Xenobots, like everything sphericalizes. So even just getting MomBot to manipulate them to try and push it away from sphere, and then how far away from sphere can you go? Clearly, you can go quite a ways. So just if that's just the yardstick, just how far can you go, right? So assuming Thomas wants to continue on in this direction, he's going to have to complicate this method. Oh, he's there.
Ah, there I am.
There you are.
Okay. Sorry, you were talking about me behind my back, Josh?
We were, and then yes. Sorry, Doug. Go ahead. You could ask your question again, or propose.
Oh, I was saying for the downstream project, or I don't remember
no,
my
just
question.
for seeing whether you can get something other than what's in the literature.
Oh yeah, so I was saying, Thomas, one thing we could do is so the keyword I would look at for this some of the stuff would be like gastruloid. So that's the name of these Xenopus explants that elongate. They essentially, so gastrulation is the process of making the gastrointestinal tract. It's the long tube. It's how you elongate. And there are ways to make these animal caps elongate or not elongate. That's been published. I think what would be super cool is to mine some of that literature or take examples that we can make and create like a state space using your tool and see, is the stuff that we can make through pies injections or other manipulations, like does it exist outside of the state space of what's been reported, right? Like, are we creating anatomies and geometries that are significantly different than we might expect from published literature, because I think that would be super cool. And that's something that downstream we could leverage to create arbitrary geometries however we want.
Yeah, it sounds sort of like, have you anyone run into this idea of a morphospace? This
Yes. Yep.
classic in biology. Yeah. Yeah, sort
Yeah.
of.
We use
you know,
what we use like morphometric programs for that, where it like morphs state space and creates PCA analyses to give you shape space.
cool. Yeah, we'd have to get, I think, a little creative on how we quantify, the state space. Like entropy is just a scalar value. There's not that much richness there. But I mean, I'm sure there's many different signals we could extract even just from the masks of the perimeter.
Yeah.
Yeah. So yeah, totally down with that. Gastroloids.
And I also wonder, I think one thing that would be really cool if we could do in future to get data like that is to have some sort of a 3D information about the bots. Like right now, we always have just one camera looking from the top. If we could have two cameras or a camera and a mirror or something like that to get the Z information, especially for those pretzel looking stuff that Doug just showed, that could be very interesting to have some 3D shape characterization.
So I don't know if it was I think it was in X1, the first Xenobots paper. There's a supplemental figure where we took images from each orientation of the Xenobots and then mapped Hausdorff distances on sort of the 3D representation to compare it to the simulated versions. It was pretty messy, so I'm not saying that's a great idea, but it might be something we could look at. Could that be adapted? We basically did like XYZ, then the diagonals, just individual images, and tried to recapitulate some sort of a 3D structure from that.
So in this, if you had some kind of 3D, like a convex hull, represented in like as some kind of 3D tensor, you could easily adapt this method. Like you're just doing it's literally just the distribution of the distances from every pixel to the centroid of the object. So we could go as many dimensions. We could do 5-dimensional objects if we felt so inclined.
Yeah, so I was just gonna say on the bio end, one way we could do that that's not awful is you can just do Z stacks. So you create a 3D model by slicing and you just use a fluorescent signal. And that would require us to probably do like it upright and flip it upside down and then paste the two together. But we could get like a volumetric 3D representation. Wouldn't be easy to do on all of them, but I think generating enough data to see if this would be worth pursuing wouldn't be too tough.
Anyone else want to jump in?
Yeah, I can go next. If
Go
you
for it.
Yep. So yeah, so here we see multiple parallel instances of self-organization going on. So the question is, can an AI learn the state variables on these series of individual instances of the self-organization rather than learning it on the complete parallel instances of self-organization? So that's the question. I want to answer that by extracting those individual instances. So why don't we focus on one particular, say, region and then extract this particular instance of these cells interacting? It's not obvious that, I mean, there are multiple things going on in this box, so it's hard to get clean data of just those particular cells that participate in the mechanical assembly of these xenobots. So what's the answer? How do we extract those individual cells? To do that, so you could run the video through CellPose, get the mask, and you can extract the trajectories of these detected cells, and these are centroids of these different clusters. And the clusters themselves, they disassemble and assemble, and there's a lot of interaction. And how do you track this information? It's hard to get that information just out of this video naively, right? So how do you extract that particular information? So to do that, we developed this pipeline where, I mean, this is just an example of cells aggregating, and here you have 1, 9, and 4 participating in that aggregation process, and some of them don't even contribute. So you can build this graph over time of how these cells interact and just extract the video that correspond to those spatial positions. So this is how you can extract this directed acyclic graph to train an AI on these different examples and see whether it predicts better. So when it predicts better, I'm talking about long-term prediction, and that would indicate that it has learned the state space, the state variables, and we can believe that it means something. So that's sort of the direction that we're going with. And this is focused towards a particular paper that we're planning to publish. Yeah.
So Doug, we're hoping we can rope you in for the preparation of this paper.
Yep, whatever you need.
Awesome. Okay.
Cool. Any questions? All right.
Is it possible that the best, just watching the video, again for the millionth time, is it possible that just like the best model might be just gravity, or that these things kind of just behave like particles that come together and stick, right? Like, because there's a lot of effort, I know you've put in, Krishna, into the machine learning thing, but I'm wondering, like, what is the simplest viable model that produces, this sort of aggregation effect?
Right. Instead of like iterating over like what's the simplest model, I mean, we need to find the simplest model directly from the data set, right? It's not obvious when you formulate the simplest model of, let's say, gravity interacting. How can you make a comparison? Like, it's hard to make a comparison against a real data set that we have.
Yeah, that's fair. I'm just rambling.
So I think this is a really good question, Thomas, because I mean, this has come up a bunch of times in these meetings. Like if we put a whole bunch of magnets on a table and shook them, and you saw the same thing, you get self-organized, the magnets all glomming together, and then you run Krishna's data video to state variable model, does it produce a model, the same model? And if it does, then that would be evidence that the cells aren't doing anything more fancy than moving around and sticking to one another. But if there is a difference among the models, that means the cells are doing something more than just passive magnets or droplets or whatever other inorganic self-assembling system you want to grab.
Right. The complexity of the state space that describes the dynamics, that's one measure where you could compare droplets, magnets or droplets versus these xenobots. I mean, Doug, you shared a video of these droplets last meeting, and I could take a look into the ways that they simulate. I think they provided details of the simulation, and you could have a model and then compare what the AI tells us regarding the state variables in that case versus the xenobots.
You could do some kind of really like kind of hacky estimate of like Kolmogorov complexity, where you say like, okay, let's say I want to get 90%, reconstruction or 90% prediction, for xenobots, magnets, and oil drops, and then just ask like, what's the minimal dimensionality of the latent vector that recovers, 90% prediction accuracy, right? That's kind of, with a little bit of hand waving, like the key complexity, isn't it? Like the smallest program that could generate your data.
Right.
Yeah. That could be fun.
Yeah. Interesting.
This is looking really far ahead, but if we did this admittedly big study already to be able to say, this thing automatically learns a model that distinguishes between biological cellular self-organization and inorganic self-organization. Just saying there's a reliable, repeatable difference that we can detect, that'd be a big deal. If we could, because this video to latent vector model is generative, you can run it and say, show me, generate, hallucinate a video of when the cells behave least like the magnets. So we, maybe 90% of the time the cells are just doing something that's, well, I don't know, whatever, not based on anything internal to the cell, just its external adhesity. So then it could start to show examples of when the cells are really, doing their thing. And maybe that would help guide the Tufts folks to like new hypotheses and new things to look for. That could be super cool. Lot of ifs, but
Yep.
anyone else with updates today?
I only just wanted to note, I will show the data next time, but just for Josh's information, or maybe the group, that one of the goals we want to do is add more sensing / response capabilities to the bots. And part of the upcoming proposal is to look at the response to pathogens and small molecules in the environment. And so I have a new student working for me. He'll be here for the next 2 months only. He's leaving in the summer, but we've already gotten some good data where it looks like we have 2 different isolates of bacterial surface markers that you can throw on the frogs and the xenobots. And so far, even though they're both from E. coli, they're just different strains, we are seeing a differential response. We're seeing a stronger response from one of the strains than the other. And this is with a really simple measure. So this week we're doing triplicates just to make sure it's real. It's only an N of 1. But what we want to do is sort of come up with a set of variables we can measure from the environment to try to get predictions about what the xenobots have seen or done. And once we get there, then we can start asking cool questions about, like, how long does it take for the response to come up, right? If a xenobot sees something, how fast can we detect that it's seen it? How fast can it tell us? And once that thing is gone, how long does the signal stay? So that's the latency. And I think we can start to survey this whole space and expand it beyond just immunity to other types of things as well. So I'm also going to be ordering some transgenic lines that turn on fluorescent reporters instead of releasing stuff into the environment. And then you can just say, how long after detecting this substance X before your xenobots start glowing green? And how long do they stay glowing green afterwards? And we may even be able to tighter those variables. So stay tuned, but it looks like preliminary wise, we already have good data, which makes me a little bit more confident in the work that we pitched, which is good.
That's good, because there's only $7 million riding on the fact.
Exactly.
I promise that
I want to jump as far ahead as we
Well done.
can so that we can sort of trickle stream the data, so that if we do have a light report, we can sort of backfill based on existing pilot work.
I've got my poker face on, but my stress levels just went down 4 notches. Thank you, Doug.
Good.
Super exciting. Wow. Okay, lots of questions. So if you get these to fluoresce, do you think there's any chance that you could do almost like the xenobits thing? So if you just did, a sprinkle of a whole bunch of self-assembling cells, and then we could say, does a 3-cell assembly fluoresce a little bit longer or brighter than a 4-cell assembly? Or is that like,
Yes.
Yeah, that's a possibility.
Yeah, I think the stuff that this dovetails with a lot of things that Mike's more interested in, but I just mean his wheelhouse, things he's already investigating, but I think we can pull this together. So yes, does the size of the unit or composition of the bot affect its response? Number 2, can we train it? So if you prime the system with a response, does it turn on faster or slower with repeated exposure, or does it maintain the signal shorter or longer? Like, that's super cool.
Yeah.
And then also the thing that I'm interested in is, can the signal be transmitted? So if you have a bunch of green bots that are secreting something, can they turn on a green signal for, like, can the swarm propagate something so that even if an individual agent has never seen the stimulus, it can receive feedback from its neighbors and pass that on in some measurable way.
Yeah. Wow. Very cool.
So yeah, I think there will be plenty to do once we get the system up and running.
Sure.
I'm just exploring the state space right now.
Yes. Well, just to know that it's possible is extremely exciting. Very, very cool, Doug. So because this is bacteria, does this dovetail at all, Elena, with your project, or are these kind of orthogonal things?
Yeah, I think it has connections, but we're trying to separate these projects
Got it.
in space. Yeah, maybe I can show you something about bacteria. Also started to talk about them. Yeah, so can you see my screen?
Yes.
Looks good.
Yeah, okay, cool. Yeah, so I established positive and negative controls for our TMRM sensor. So TMRM sensor is a membrane permanent cationic fluorescent dye to detect membrane potential in bacterial cells. As a positive control, we use citrate, which produces hyperpolarization of bacterial cells. And as negative control, which I didn't show last time when we met with Guru. Yeah, so as a negative control, we use CCCP, which is a protonophore, and it's also uncoupler of oxidative phosphorylation. Yeah, so basically what we expect to see, that bacteria will fire up in response on citrate, and the signal will be gone in response to CCCP. Yeah, and so please focus on the left frame. This will be video of about 2 hours long. At this time, I will add citrate. You will see that when screen turns black, that means I open microscope and I pipette the chemicals. Then positive reaction will develop, and about this time, I will add CCCP and reaction will go. Yeah, so, and on the right side is a control. So the same bacteria, the same sensor, but instead of citrate and CCCP, I added just bacterial media. Yeah, so I'm pulling that. Yeah, so finally, when positive and negative controls confirm that TMRM dye works in my hands, we can combine this culture. This is liquid culture. We can combine it with xenobots, and I already started to do that. And I recorded long time lapse video over weekend. It was like almost 4 days video. Yeah, so, and I see some interesting things, how bacteria react on xenobots staying there. And they have some differences between younger and older bots. Younger, they don't have cilia, they don't move. Older bots, they have cilia, they try to move. Yeah, so it's just a teaser at this point. I will show this data next time. Yeah.
And Elena, sorry, just to pull back a little bit. So the goal here for this part is to just make sure that the xenobots and the bacteria can cohabitate, right?
At this point, the idea, so we use this TMRM sensor to record any electrochemical reaction from bacteria. So if they change membrane potential, we should be able to see that with TMRM dye under microscope. Yeah, before I didn't see any perturbations of TMRM when I put bots into the bacterial culture, and it was worrisome. Like, we didn't know, xenobots didn't produce any reaction, or TMRM dye doesn't work, or bacteria don't react. So it was questionable. And then we just went back to very beginning and started to establish clear positive and negative controls just to make sure that our system works. Yeah, and this is what I have done and showed now that TMRM dye works. So if xenobots produce any reaction in bacterial cells, we should be able to see that.
Got it. Got it.
Elena, did you ever send me the photos of the with Xenobots and without Xenobots.
Yes, what I sent you at that time, the conditions for TMRM sensor were not optimal. So
Okay.
it's good that we didn't jump to many data and to analysis, because now we have much better conditions, and now TMRM dye finally works. Yeah, so I will generate data and send you something.
Cool. Yeah.
Yeah, because I definitely see differences in reaction. Like when I put bots, I see some bacterial response.
Yeah, it would be great to try to quantify that a little bit. And
Yeah.
Elena, can you maybe check the only thing I'm still like trying to wrap my head around the project is the with normal gram-negative bacteria, it will stimulate the innate immune response, and they will epithelia will hemorrhage out or release antimicrobial peptides, which is sort of what I'm trying to look at for basically just killing stuff. And so I'm just wondering, when you look at communication, if they're just like poisoning the bacteria, that's communication in one aspect, but you may just be getting like it's like dumping acid on something, right? Like the question is sort of, is it meaningful? So I also wonder, could you culture a bunch of xenobots in a small volume of media and then like filter the xenobots out and just put the xenobot media into the bacteria so that you get all of the stuff that they've been releasing, and you can see if there's any response, like minus xenobot but plus xenobot product, where the two have never really seen each other.
Yeah, yeah, that's reasonable, definitely.
So if you dropped acid on something and you saw changes in all of the material that's in contact with the acid, that's uninteresting. But if things beyond the periphery of where the acid is start changing, that's the things that are being hit with the acid communicating, saying. So Thomas, some of your analysis could be useful here, again, looking for info flows beyond, where there's actual physical contact with the media.
yeah, absolutely.
Cool. Thanks, Elena. Anyone else? Okay. If not, thanks everyone. Thanks for the updates. I'll circulate the video. And again, my apologies about starting late. So we'll meet 2 weeks from now at 12:30.
One last note, Caitlin, before I lose you. I've checked on the paper a bunch of times. I've not seen any edits from Sarah Walker, so I will poke her again gently this week so that we can keep making progress.
Okay. Yeah, that sounds good. I was
Okay.
wondering about that. Thanks.
Do you want me to poke her as well?
Sure. You could just say, do you have, maybe just say like, can we come up with a timeline would be useful.
Okay. I'll do that now. Okay. Thanks everyone.